Welcome to Lecture 17, where we will go over the processes of osmoregulation and the excretory system. A common theme in organisms in the regulation of water concentrations is that they typically regulate or transport salts or other ions across the membrane. In doing so, they can direct the flow of water in and out of tissues as well as vessels. Take a look at the diagram below to refresh your memory with regards to what a hyperosmotic, isoosmotic, and hypoosmotic solution or conditions are. This will help you understand the movement of solutes as well as water across a membrane. When we first refer to the absolute movement of just water molecules, remember this is what we term osmosis. Osmoregulation and excretion are typically discussed together because both of these processes are involved in homeostasis. However, they refer to two different things. Osmoregulation refers to the ability of an organism to regulate solute concentrations as well as water gain and loss, whereas excretion refers to the ability of an organism to eliminate metabolic waste products such as urea from the body. Osmolarity is a measure given to a solute concentration. It is typically expressed in number of moles of solute or number of milliosmoles per liter of solution. Blood, for instance, has a typical osmolarity value of about 300 milliosmoles per liter, while seawater has a much higher value of about 1,000. Animals can maintain water balance in two main ways. Most marine animals are termed osmoconformers. This means that they maintain themselves in an isoosmotic condition with their environment. Freshwater and terrestrial animals, however, are osmoregulators. This means that they must maintain an internal osmotic state despite the varying external environment around them. We're first going to take a look at how marine animals osmoregulate. Most marine animals, as stated in the previous slide, are osmoconformers. This means that they have the same general osmolarity value within their tissues as the seawater or surrounding environment. However, they still have a problem. They must maintain lower or higher levels of specific solutes such as magnesium as well as calcium. As a result, some marine fish, like cod, constantly drink in seawater. This is to offset the fact that water is constantly being lost from the body through osmosis. Excess salts can then be excreted through the gills and the kidneys. Other marine fish, such as sharks, keep higher salt concentrations in their tissues. Thus, there's excess water that is constantly entering into the body through osmosis. How they get around this fact is that they can excrete much of the excess water through their own urine. The diagram to the right gives you an idea of the pathway of water and salts in a typical marine fish. Freshwater animals, on the other hand, are osmoregulators and are typically kept hyperosmotic to their environment. This means that water from their surrounding environment will constantly pass into them through osmosis. As a result, there are many accommodations that these organisms have. For instance, freshwater fish, such as perch, can excrete highly diluted urine to accommodate for this excess water. They replenish salts from food and by uptaking salts from across the gills as water passes over. Migratory fish, like salmon, experience both the terrestrial as well as marine environment. However, when they're in a freshwater environment, like perch, they can excrete highly diluted urine. When they migrate into marine waters, they produce high levels of cortisol. This, in turn, will increase the number and size of salt-secreting cells, and also allows them to excrete excess salt from the gills. This allows a switch in the urine production for them to produce small amounts of concentrated urine when in a marine environment. Take a look at the diagram below and you will see the pathway of water and salts in a typical freshwater fish. Some animals are highly adapted to live in temporary waters, which means they must be adapted to survive periods of dehydration. Many of these animals, such as the tardigrade, can enter into dormant periods that are called anhydrobiosis. This means that they can lose nearly all of their body water and still survive. The diagram below shows a difference between a hydrated and dehydrated tardigrade. These are also called water bears. Tardigrades can enter into a severely dehydrated state that can last decades. They can also become reactivated simply with the contact of water. This is a very important process to understand, as it may hold the key to preserving other biological materials. Terrestrial animals also show adaptations for osmoregulation. In dry terrestrial environments, animals need to conserve water and also to prevent desiccation. Water is constantly being lost through the skin, the respiratory surfaces, in urine, as well as in the production of feces. Some of these adaptations include having nocturnal activities to avoid the high heats and low humidity conditions, the consumption of moist food and the ability to extract the most moisture from the food as possible, and also many of these organisms have a much higher heat tolerance. Thus, they have a reduction in the amount of sweat they need to produce in order to conserve water. Camels, for instance, have a higher core body temperature and also can withstand periods of extreme water loss. Many animals have what is termed specialized epithelial layers, called transport epithelia. This transport epithelia can move solutes in a specific direction, whether it be active or passive transport. Transport epithelia is usually arranged in tubular networks with a very large surface area. This increases the amount of surface area available to control the movement of ions in water. This is very important in helping to regulate the solute concentrations in blood or in the surrounding hemolymph. Ultimately, this is for osmoregulation. For example, the albatross is a marine bird that has salt glands that are lined in transport epithelium in the nasal area. These salt glands can excrete a salt solution in order to prevent dehydration in a marine environment. The other diagram to the right also shows the distribution of transport epithelium and the movement between blood flow and salt secretion in the nasal passageways. Excretion involves the process of the body eliminating ammonia and other metabolic waste. Keep in mind that proteins and nucleic acids both have amino groups, and these amino groups are typically converted into a form of nitrogenous waste to be eliminated from the body. The three main types of nitrogenous waste in animals include ammonia, urea, as well as uric acid. Ammonia includes NH3 as well as the ammonium ion NH4+. Ammonia is considered to be very toxic even at low concentrations. As a result, this is present in most aquatic animals because animals, in order to produce ammonia, need to access high volumes of water. Urea, on the other hand, is produced from NH3 and carbon dioxide, CO2. Urea has a lower toxicity value and is also considered to be highly soluble. However, it is energetically costly to produce. Mammals, most amphibians, sharks, and bony fishes will produce urea as their metabolic waste product. Uric acid, lastly, consists of NH3 as well as NH4+. It is also arranged in a ring and more stable form. It is considered to be non-toxic, but has a very low solubility value. As a result, it is typically excreted in paste form. This is even more costly to produce than urea. Uric acid is the waste form produced by most birds, reptiles, insects, and terrestrial snails. Take a look at this diagram to understand how nucleic acids and proteins contribute to the formation of amino groups that then need to be excreted in the multiple waste forms. The type of waste excreted by an animal is highly dependent upon their environment, the energetic costs of producing the product, the toxicity of the product, as well as the diet of what is being consumed. For instance, many predators excrete more nitrogen due to their consumption of other animal tissues, and aquatic environments allow for the production of ammonia because it can dissipate out more of the toxic waste. While having on the organism producing the waste. We're now going to take a look at the basic steps involved in the functioning of the excretory system. There are four main processes that occur: filtration, reabsorption, secretion, and excretion. Many of the excretory systems involve a tubular network that is held in close association with the capillary. Filtration involves high pressure in the blood, which causes water and solutes to leave the blood and are thus collected within the tubule. Reabsorption refers to the fact that important ions and other substances need to be returned to the blood. This is typically carried out by the transport epithelium. Secretion refers to the elimination of excess wastes and toxins that are extracted from the body fluids. These wastes and toxins will be deposited within the center lumen of the tubule. And then finally, excretion refers to the elimination of the filtrate or urine as it leaves the body. There are many different forms of these tubular networks that assist in excretion of materials. These include protonephridia, metanephridia, Malpighian tubules, as well as the typical kidney. We're now going to take a look at each of these excretory systems. The protonephridia consists of a network of tubules that are capped by what is termed flame bulbs. The flame bulbs are important in the filtration process. Flame bulbs consist of a tubule cell, a cap cell, as well as a tuft of cilia. The protonephridia can also be adapted for osmoregulation as well as nitrogenous waste removal. Protonephridia are present in worms such as Platyhelminthes, rotifers, some annelids, lancelets, as well as some mollusk larvae. With respect to filtration, the cilia will draw the water in, as well as other types of solutes from the interstitial fluid that is bathing the flame bulb. This will direct the filtrate then into the tubule. The filtered urine then exits the body, typically through an opening in the body wall or external pores. These can be found throughout the body of the organism. Metanephridia are a bit more complex than the protonephridia and consist of a network of tubules and capillaries that are typically housed in the coeloma cavity. In earthworms, the metanephridia is important in producing hypoosmotic urine. This helps to balance the high influx of water that is constantly entering the animal through the skin. Cilia will help to direct the coelomic fluid into the internal opening. Refer to the diagram to the left. The coelomic fluid will be directed through ciliary movement. Once the coelomic fluid enters into the tubules, it will then go through the network of tubules. Transport epithelium that lines these tubules is important in reabsorbing most of the solutes and also in retaining the nitrogenous wastes and keeping them within the lumen of the tubule to be excreted. The coelomic fluid will be filtered as it moves through the capillary and the tubule network. It will then be collected in the storage bladder for elimination out the external opening. Note that each segment of the earthworm will have a metanephridia present to help eliminate out waste throughout the body of the animal. Malpighian tubules are commonly found in insects, as well as other types of terrestrial arthropods. Malpighian tubules consist of blind-ended tubules that extend out from the digestive cavity. These tubules are then bathed in the hemolymph of the animal. Malpighian tubules are important in aiding in nitrogenous waste removal as well as osmoregulation. Malpighian tubules are highly efficient at conserving water. Thus, they are considered to be an adaptation and highly beneficial for terrestrial living. In the Malpighian tubule, transport epithelium will help to secrete nitrogenous waste and remove the nitrogenous waste and various solutes from the hemolymph. These nitrogenous waste and solutes will collect into the lumen of the Malpighian tubule. Water then enters the tubule through osmosis. The water and solutes are then reabsorbed as nitrogenous waste and form uric acid as it enters into the rectum. This will then be excreted along with the feces. Note note that there is no filtration in this process. We're now going to take a look at the structure and function of the kidney. The kidney is a specialized organ that is common in vertebrates as well as some chordates. It contains highly arranged tubules and capillary networks called nephrons. These will help in excretion as well as osmoregulation. When we take a look at the filtration process, the nephron is responsible for filtering out salts, glucose, amino acid, vitamins, nitrogenous waste, and many other molecules from the blood. This filtrate will then eventually collect into the pelvis of the kidney. This will then travel through the ureter and then travel and be stored in the bladder. This will then be eliminated out of the body through the urethra. Take a look at the diagram to the right to understand the overall structure of the kidney. Note that we have the outer layer, which is termed the renal cortex, which surrounds the more inner layer, the renal medulla. Also note that there is a renal artery and renal vein that are bringing blood to and from the structure. Also note that coming from the renal pelvis, where most of the urine that is produced will collect, will then empty into the ureter. This will eventually lead to the bladder and then to the urethra.